Radiolabeling techniques, detailed lecture. Radiolabeling refers to the technique of incorporating radioactive isotopes, radioisotopes, into molecules to track or quantify biological processes with high sensitivity. These isotopes serve as tracers because their radiation emissions can be detected with high precision. Radiolabeling is a fundamental tool in molecular biology, biochemistry, physiology, ecology, and pharmacokinetics. It allows scientists to follow the path, localization, uptake, metabolism, and interactions of labeled molecules in living systems or in vitro setups. Radioisotopes used in radiolabeling emit different types of radiation: alpha, alpha, beta, beta, or gamma, gamma rays. Among these, beta emitters such as cubed H tritium to the power of 4C carbon 14 cube squared P phosphorus 32 and cube to the power of 5S SULFUR 35 are commonly used in biological labeling because they provide stable incorporation into biomolecules like DNA, RNA, proteins, and metabolites. The process of radiolabeling involves either chemical synthesis, where radioactive atoms are incorporated during molecule formation, or enzymatic labeling, where enzymes transfer radioactive groups to specific substrates. For example, cube squared P labeled ATP can be used to track phosphorylation events in protein signaling pathways, and to the power of 4C labeled glucose can be used to trace metabolic pathways like glycolysis in the Krebs cycle. In molecular biology, radiolabeled nucleotides are used in techniques such as northern and southern blotting, DNA footprinting, in situ hybridization, and DNA sequencing, where radioactive probes allow detection of specific nucleic acid sequences with autoradiography. In ecology and physiology, radiolabels are used to track nutrient flow in food chains, hormone transport, or the uptake of nutrients and toxins in organisms. Radiolabeling has also been extensively used in pharmacokinetics, where drugs labeled with radioisotopes are administered to animals or humans, and their distribution, absorption, metabolism, and excretion ADME are tracked. This provides quantitative data on drug behavior over time. The detection of radiolabeled molecules is typically done using instruments such as Geiger counters, liquid scintillation counters, and autoradiography. Autoradiography involves placing radiolabeled samples on photographic film or phosphor screens, where emitted radiation creates an image corresponding to the location of the labeled molecule. Advantages of radiolabeling include extremely high sensitivity, specificity, and the ability to trace molecules in complex biological systems without disturbing their function. However, the handling of radioactive materials requires strict safety protocols due to potential health hazards from radiation exposure. Proper shielding, waste disposal, and licensing are essential in labs using radioisotopes. Short-lived isotopes, e.g., cube squared P, with a half-life of approximately 14 days, are often preferred for short-term experiments, whereas long-lived isotopes like to the power of 4C, with a half-life of approximately 5,700 years, are used in long-term labeling studies or for carbon dating. Biological examples. Cube squared P DNA labeling to detect DNA synthesis rates or to study DNA-protein interactions. To the power of 4C labeled amino acids to measure protein synthesis. Cube H thymidine incorporation to assess cell proliferation in tissue culture. Radiolabeled hormones, e.g., squared to the power of 5I insulin to study receptor binding and signal transduction. Practice MCQs. Q1, which of the following radioisotopes is most commonly used to label DNA in molecular biology? A to the power of 4C. B cubed squared PC cubed to the power of 5SD squared to the power of 5I answer, B cubed squared B. Q2, what is the purpose of using radiolabeled compounds in metabolic studies? A to increase reaction rates. B to visualize and quantify molecular movement. C to reduce metabolic activity. D to enhance enzyme activity. Answer, B to visualize and quantify molecular movement. Let me know if you'd like the next lecture on chromatography techniques, electrophoresis, or spectroscopy methods in biological sciences. You said. Detection and measurement of different types of radioisotopes normally used in biology. ChatGPT said. Here's a detailed, paragraph-style lecture on detection and measurement of different types of radioisotopes normally used in biology, suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology. Detection and measurement of radioisotopes in biology, detailed lecture. Radioisotopes are widely used in biological sciences for tracing biochemical pathways, studying molecular interactions, and quantifying physiological processes. Their utility lies in their ability to emit detectable radiation. To harness their potential, one must understand how to detect and measure the type and intensity of radiation emitted by different radioisotopes. The choice of detection method depends on the type of radiation, alpha, beta, or gamma energy level, half-life, and the biological application. In biological research, the most commonly used radioisotopes include tritium cubed H, a low-energy beta emitter, used to label DNA, RNA, or proteins. Carbon 14 to the power of 4C, a beta emitter, often used in metabolic studies. Phosphorus 32 cubed squared P, a high-energy beta emitter, frequently used to label DNA or nucleotides. Sulfur 35 cubed to the power of 5S, a beta emitter, used in protein and amino acid labeling. Iodine 125 squared to the power of 5I, a low-energy tritium cubed H, a low-energy beta emitter, used to label DNA, RNA, or proteins. Carbon 14 to the power of 4C, a beta emitter, often used in metabolic studies. Phosphorus 32 cubed squared P, a high-energy beta emitter, frequently used to label DNA or nucleotides. Sulfur 35 cubed to the power of 5S, a beta emitter, used in protein and amino acid labeling. Iodine 125 squared to the power of 5I, a low-energy gamma emitter, used for hormone and receptor binding studies. Detection instruments and techniques. Geiger-Müller GM counter. The GM counter detects ionizing radiation by producing a current pulse when radiation passes through a gas-filled tube. It is effective for detecting beta and gamma radiation, especially from isotopes like cube squared P and squared to the power of 5I. However, it lacks the ability to distinguish between different isotopes or quantify energy levels accurately. Liquid scintillation counter LSC. LSC is highly sensitive and widely used to measure low-energy beta emitters, such as cubed H into the power of 4C. In this method, a radiolabeled sample is mixed with a scintillation cocktail that emits light photons when beta particles interact with it. These photons are then detected and quantified by photomultiplier tubes. This method is especially valuable in detecting weak beta emitters that cannot be picked up by GM counters. Gamma counter. Gamma counters use sodium iodide night scintillation detectors to measure gamma rays, especially from isotopes like squared to the power of 5I. This is essential in studies involving radiolabeled hormones or ligands, where gamma radiation penetrates tissue and can be quantified with precision. Autoradiography. This technique uses photographic film or phosphor imaging plates to detect radioisotopes. The radiolabeled biological samples are placed against the film, and the emitted radiation creates a latent image, which is developed to visualize the distribution of the isotope. Autoradiography is widely used in molecular biology, e.g., southern northern blotting and histological localization of radioisotopes. Solid scintillation counting, dry counting. For samples on solid supports like filters or membranes, e.g., in blotting experiments, solid scintillation counters can be used. Detection instruments and techniques. Geiger-Müller GM counter. The GM counter detects ionizing radiation by producing a current pulse when radiation passes through a gas-filled tube. It is effective for detecting beta and gamma radiation, especially from isotopes like cube squared p and squared to the power of 5i. However, it lacks the ability to distinguish between different isotopes or quantify energy levels accurately. Liquid scintillation counter LSC. LSC is highly sensitive and widely used to measure low-energy beta emitters, such as cube H to the power of 4c. In this method, a radiolabeled sample is mixed with a scintillation cocktail that emits light photons when beta particles interact with it. These photons are then detected and quantified by photomultiplier tubes. This method is especially valuable in detecting weak beta emitters that cannot be picked up by GM counters. Gamma counter. Gamma counters use sodium iodide night scintillation detectors to measure gamma rays, especially from isotopes like squared to the power of 5i. This is essential in studies involving radiolabeled hormones or ligands, where gamma radiation penetrates tissue and can be quantified with precision. Autoradiography. This technique uses photographic film or phosphor imaging plates to detect radioisotopes. The radiolabeled biological samples are placed against the film, and the emitted radiation creates a latent image, which is developed to visualize the distribution of the isotope. Autoradiography is widely used in molecular biology, e.g., southern northern blotting and histological localization of radioisotopes. Solid scintillation counting, dry counting. For samples on solid supports like filters or membranes, e.g., in blotting experiments, solid scintillation counters can be used. These utilize phosphor screens or detectors placed directly on the sample. Cherenkov counting. A specialized form of detection for high-energy beta emitters like cube squared p, where radiation exceeds the speed of light in a medium and produces visible light. This method is useful for counting without adding scintillation fluid, though it's less sensitive than traditional LSC. Measurement parameters. Counts per minute CPM, the raw output of detected disintegrations. Disintegrations per minute DPM, the actual number of radioactive decays, calculated by correcting CPM for detector efficiency. Specific activity, radioactivity per unit mass of a compound, useful for comparing labeling efficiency. Half-life T1/2, the time required for half the atoms of a radioisotope to decay, important for experiment planning and waste management. Biological importance and safety. Detection of radioisotopes allows biologists to quantify dynamic processes such as DNA replication, protein synthesis, ligand receptor binding, and metabolic flux. However, radiation safety protocols are essential to minimize exposure. Shielding plexiglass for beta, lead for gamma, proper waste disposal, and radiation badges for monitoring exposure are standard in all radiological labs. Practice MCQs. Q1. Which of the following radioisotopes requires a liquid scintillation counter for accurate detection due to its low energy beta emissions? A. Cubed squared p. B. Squared to the power of 5i c cubed hd to the power of 6 county answer, c cubed h. Q2. Gamma radiation emitted by squared to the power of 5i is best detected using a Geiger counter. B. Autoradiography C. Gamma counter with night detector. D. Liquid scintillation counter answer, C. Gamma counter with night detector. Let me know if you want the next topic on RFLP, PCR, or ELISA. You said. Incorporation of radioisotopes in biological tissues and cells, ChatGPT said. Here's a detailed lecture on the incorporation of radioisotopes in biological tissues and cells, written in a paragraph style suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology and biology. Incorporation of radioisotopes in biological tissues and cells, detailed lecture. The incorporation of radioisotopes into biological tissues and cells is a critical technique in experimental biology, used to trace, measure, and visualize dynamic biochemical processes with high precision. This method involves introducing radioactive atoms into biologically relevant molecules, such as nucleotides, amino acids, sugars, or hormones, which then get metabolically or structurally incorporated into the cells or tissues of interest. The labeled molecules, now radioactive, can be followed through various metabolic pathways, enabling researchers to study gene expression, cell proliferation, protein synthesis, nutrient uptake, or signal transduction events. The most common method of incorporation is metabolic labeling, where radioisotope labeled precursors are supplied to living cells or tissues in culture or to whole organisms. For example, cubed H thymidine, tritiated thymidine, is incorporated into newly synthesized DNA during the S phase of the cell cycle and is widely used in cell proliferation studies. Similarly, cubed to the power of 5s labeled methionine or cysteine can be added to the medium to label newly synthesized proteins, as sulfur is found in these amino acids. To the power of 4c labeled glucose is used to trace glycolytic flux or respiration, while cubed squared p labeled phosphate can be incorporated into ATP and nucleotides, aiding in the study of phosphorylation events and nucleotide turnover. Once incorporated, these labeled biomolecules can be detected using techniques like autoradiography, liquid scintillation counting, or gamma counting, depending on the nature of the isotope. For tissue-level studies, autoradiographic techniques allow for the spatial visualization of isotope incorporation at the cellular or subcellular level using photographic film or phosphor screens. This has been extremely valuable in developmental biology and histological studies to localize gene activity or nutrient transport. In plant biology, radioisotopes like cubed squared p to the power of 4c are supplied through the roots or leaves and are then tracked throughout the vascular system to study nutrient transport, photosynthetic assimilation, or hormone movement. In animal systems, isotopes can be introduced via injection, ingestion, or inhalation, and tissues can be harvested after set intervals to determine uptake and incorporation. The efficiency of incorporation depends on several factors: the metabolic activity of the cells, the concentration and specific activity of the radiolabeled compound, incubation time, and cellular uptake mechanisms. Pulse chase experiments are often used to understand the temporal dynamics of incorporation. In such experiments, cells are briefly exposed to the radiolabeled compound pulse followed by a non-radioactive version, chase, allowing researchers to study the turnover or processing of biomolecules over time. Safety considerations are crucial during incorporation experiments. For example, cubed squared p, a high-energy beta emitter, requires protective shielding to prevent radiation exposure, whereas low-energy beta emitters like cubed H or to the power of 4c pose less risk but still require proper handling and waste disposal. The radioactive materials used must be bioavailable, stable under experimental conditions, and should not disrupt normal cellular function. Examples in biology. Cubed squared P orthophosphate incorporation in phosphorylation studies of proteins. Cubed to the power of 5s methionine for studying protein translation in cell-free systems. Cubed H uridine incorporation to study RNA synthesis in cultured cells. To the power of 4C labeled sucrose to trace phloem loading and translocation in plants. Practice MCQs. Q1. Which of the following radioisotopes is commonly incorporated into DNA during cell proliferation assays? A. Cubed squared P. B. Cubed to the power of 5s. C. Cubed H thymidine D squared to the power of 5i. Answer. C. Cubed H thymidine. Q2. What is the primary method used to detect the location of incorporated radioisotopes within tissue sections? A. PCR. B. ELISA. C. Autoradiography. D. Mass spectrometry. Answer. C. Autoradiography. Let me know if you'd like the next lecture on isotope dilution techniques, ELISA, or immunofluorescence methods. You said, molecular imaging of radioactive materia. ChatGPT said. Here's a detailed, paragraph-style lecture on molecular imaging of radioactive material, tailored for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology and biology. Molecular imaging of radioactive material, detailed lecture. Molecular imaging is a powerful technique in biological and biomedical research that allows for the non-invasive visualization, characterization, and quantification of biological processes at the molecular and cellular levels in living organisms. When radioactive materials, radioisotopes, are used in molecular imaging, they serve as tracers that emit radiation detectable by sensitive instruments, allowing scientists to monitor specific biochemical events in real time within intact tissues or whole organisms. This form of imaging bridges the gap between molecular biology and in vivo physiology, enabling dynamic observation of processes such as gene expression, cell signaling, metabolism, and drug receptor interactions. Radioactive molecular imaging techniques rely on radiotracers, radioactive compounds that are designed to bind or participate in specific biological pathways. These tracers are often labeled with gamma-emitting or positron-emitting radioisotopes. For example, fluorine-18 to the power of 8F, a positron emitter, is used in fluorodeoxyglucose (FDG), which is taken up by metabolically active cells and is widely used in positron emission tomography (PET). Likewise, technetium-99 meters to the power of 9 to the power of 9 Tc, a gamma emitter, is used in single photon emission computed tomography (SPECT). Positron emission tomography (PET) is one of the most sensitive techniques for molecular imaging. In PET, radiotracers that emit positrons undergo annihilation reactions with electrons, producing pairs of gamma photons that travel in opposite directions. Detectors arranged in a ring around the subject capture these photons and reconstruct a three-dimensional image of tracer distribution. PET is used in neuroscience to study brain function, in oncology to image tumors, and in cardiology to assess heart metabolism. Single photon emission computed tomography (SPECT) is another molecular imaging modality that uses gamma-emitting isotopes. SPECT provides information about the functional state of tissues and is useful in mapping receptor density, blood flow, or metabolic activity. Unlike PET, SPECT tracers do not require annihilation reactions, and its instruments are generally more widely available and less expensive. In molecular biology, radioactive imaging is also done using autoradiography, where radioactively labeled molecules such as DNA, RNA, or proteins are separated via gel electrophoresis or blotted onto membranes and then visualized using photographic film or phosphor screens. This static imaging is vital for gene expression studies, mutation detection, and protein DNA interaction analysis. Additionally, radio-labeled antibodies or ligands are employed in targeted imaging of specific tissues or cell types. For example, antibodies labeled with squared to the power of 5i or to the power of 9 to the power of 9 Tc can target cancer cell markers and help in the localization and diagnosis of tumors. In gene expression studies, radio-labeled nucleotide probes are used in situ hybridization to visualize mRNA expression in tissue sections. Safety and specificity are crucial when using radioactive materials for imaging. The radiotracer must exhibit high affinity, specificity, and stability, with minimal toxicity or interference in normal biological function. Proper shielding and dosimetry are required to minimize radiation exposure to both the subject and the researcher. Molecular imaging of radioactive material thus provides a dynamic, real-time view of molecular events, making it indispensable in diagnostics, drug development, functional genomics, and systems biology. Practice MCQs. Q1. Which of the following imaging techniques uses positron-emitting radioisotopes for molecular imaging? A. SPECT. B. PET. C. MRID. CORT. Answer. B. PET. Q2. What is the primary radioactive tracer used in PET imaging to detect metabolic activity in cancer cells? A. Cubed I. B. Cubed H thymidine C to the power of 9 to the power of 9 TCD to the power of 8 FFDG. Answer. D to the power of 8 FFDG. Let me know if you want the next lecture on isotope dilution, immunofluorescence methods, or enzyme-linked assays. You said, safety guidelines. ChatGPT said. Here's a detailed, paragraph-style lecture on safety guidelines in the use of radioactive materials in biology, ideal for CSIR Net Life Sciences, GATE, and PhD entrance preparation in zoology and related biological sciences *. Safety guidelines in the use of radioactive materials in biology, detailed lecture. The use of radioactive materials in biological research demands strict adherence to safety guidelines to protect both researchers and the environment from harmful exposure. Ionizing radiation emitted by radioisotopes, such as alpha, beta, and gamma rays, can damage living cells, cause DNA mutations, and increase the risk of cancer. Therefore, comprehensive radiation safety protocols have been developed to ensure safe handling, storage, and disposal of radioactive substances in laboratory and clinical settings. One of the primary principles of radiation protection is the ALARA principle, as low as reasonably achievable, which encourages minimizing exposure by optimizing three factors: time, distance, and shielding. Exposure time should be kept as short as possible, distance from radioactive sources should be maximized, and appropriate shielding like lead bricks or plexiglass barriers should be used depending on the type of radiation emitted. For example, plexiglass is commonly used to shield beta emitters like cubed squared P, while lead shielding is necessary for gamma emitters like cubed R to the power of 9 to the power of 9 Tc. Only trained personnel should handle radioactive materials, and they must follow protocols set by national regulatory bodies such as the Atomic Energy Regulatory Board (AERB) in India or radiation safety committees of respective institutions. should handle radioactive materials, and they must follow protocols set by national regulatory bodies such as the Atomic Energy Regulatory Board (AERB) in India or radiation safety committees of respective institutions. Laboratory workers must use personal protective equipment (PPE), including lab coats, gloves, and sometimes face shields. Radiation dosimeters, film badges or TLD badges, are worn to monitor cumulative radiation exposure. Laboratory areas where radioactive materials are used must be clearly marked with radioactive hazard symbols. Safety guidelines in the use of radioactive materials in biology. Detailed lecture. The use of radioactive materials in biological research demands strict adherence to safety guidelines to protect both researchers and the environment from harmful exposure. Ionizing radiation emitted by radioisotopes, such as alpha, beta, and gamma rays, can damage living cells, cause DNA mutations, and increase the risk of cancer. Therefore, comprehensive radiation safety protocols have been developed to ensure safe handling, storage, and disposal of radioactive substances in laboratory and clinical settings. One of the primary principles of radiation protection is the ALARA principle, as low as reasonably achievable, which encourages minimizing exposure by optimizing three factors: time, distance, and shielding. Exposure time should be kept as short as possible, distance from radioactive sources should be maximized, and appropriate shielding like lead bricks or plexiglass barriers should be used depending on the type of radiation emitted. For example, plexiglass is commonly used to shield beta emitters like cube squared p, while lead shielding is necessary for gamma emitters like cube r to the power of 9 to the power of 9 Tc. Only trained personnel should handle radioactive materials, and they must follow protocols set by national. One of the primary principles of radiation protection is the ALARA principle, as low as reasonably achievable, which encourages minimizing exposure by optimizing three factors: time, distance, and shielding. Exposure time should be kept as short as possible, distance from radioactive sources should be maximized, and appropriate shielding like lead bricks or plexiglass barriers should be used depending on the type of radiation emitted. For example, plexiglass is commonly used to shield beta emitters like cube squared p, while lead shielding is necessary for gamma emitters like cube r to the power of 9 to the power of 9 Tc. Only trained personnel should handle radioactive materials, and they must follow protocols set by national regulatory bodies such as the Atomic Energy Regulatory Board (AERB) in India or radiation safety committees of respective institutions. Laboratory workers must use personal protective equipment (PPE), including lab coats, gloves, and sometimes face shields. Radiation dosimeters, film badges, or TLD badges, are worn to monitor cumulative radiation exposure. Laboratory areas where radioactive materials are used must be clearly marked with radioactive hazard symbols, and access must be restricted to authorized personnel. A well-ventilated, designated radioisotope work area should be used, often equipped with fume hoods or laminar flow cabinets to prevent inhalation of radioactive aerosols. Surfaces should be non-absorbent and easy to decontaminate, and spill kits must be readily available. Regular surface contamination checks using Geiger-Müller counters or scintillation detectors help monitor cleanliness and safety. In case of spills, immediate containment, notification to safety officers, and proper cleanup procedures are mandatory. Storage of radioactive materials must be done in shielded, labeled, and secure containers, segregated according to isotope type and half-life. Logbooks should record the inventory, usage, and disposal details. Disposal of radioactive waste follows specific guidelines. Short-lived isotopes may be stored for decay, while others require authorized disposal facilities. Liquid waste should never be poured down the drain unless permitted, and solid radioactive waste must be incinerated or buried according to institutional and environmental regulations. Moreover, researchers must undergo regular training and refresher courses in radiation safety and emergency procedures. Emergency protocols include steps for contamination incidents, accidental exposure, and fire hazards involving radioactive materials. In institutions, the Radiation Safety Officer (RSO) is responsible for compliance with regulatory norms and training. In summary, safe use of radioactive materials is critical in biological research and demands rigorous attention to established protocols. These guidelines not only ensure researcher and environmental safety but also uphold the integrity and reproducibility of scientific research involving radioisotopes. Practice MCQs. Q1. What does the ALARA principle in radiation safety stand for? A. All lead and radiation approved. D. As long as radiation accumulates. C. As low as reasonably achievable. D. Allowable limits against radiation accidents. Answer. C. As low as reasonably achievable. Q2. Which shielding material is most appropriate for high-energy beta emitters like cube squared p? A. Paper. B. Lead. C. Plexiglass. D. Aluminum. Answer. C. Plexiglass. Let me know if you'd like the next lecture on ELISA, immunofluorescence, flow cytometry, or any other technique. You said. More about that topic ChatGPT said. Here's a continued and expanded lecture on safety guidelines in the use of radioactive materials in biological research, providing further insight into types of hazards, biological effects, institutional responsibilities, accident management, and international standards. Extended lecture on safety guidelines in the use of radioactive materials in biology. Types of radiation hazards. Radiation hazards are generally classified into external and internal exposure. External exposure occurs when a radioactive source is outside the body, potentially affecting skin, eyes, or deeper tissues depending on the radiation type. Internal exposure, which is more dangerous, happens when radioactive materials enter the body through inhalation, ingestion, absorption through skin, or accidental injection. For example, accidental inhalation of radioactive iodine, cubite, can localize in the thyroid and cause tissue damage. Hence, stringent protocols are followed to contain airborne isotopes and prevent internal contamination. Biological effects of radiation. Ionizing radiation interacts with biological tissues by producing free radicals, particularly through radiolysis of water, leading to DNA strand breaks, chromosomal aberrations, mutations, or cell death. Tissues with high meiotic rates, such as bone marrow or intestinal epithelium, are more vulnerable. While acute high-dose exposure may cause radiation sickness, chronic low-dose exposure increases the risk of cancer, reproductive damage, or hereditary effects. Therefore, cumulative exposure is monitored with dosimeters, and limits are set by organizations like ICRP, International Commission on Radiological Protection. Institutional responsibilities and infrastructure. Every institution using radioactive materials must maintain a radiation protection program, which includes appointment of a Radiation Safety Officer (RSO), formation of an Institutional Radiation Safety Committee (RSC), periodic risk assessments and internal audits, establishment of controlled and supervised areas with restricted access, training programs for all radiation workers, maintenance of detailed records of radioisotope procurement, use, and disposal. The AERB and India or equivalent national bodies require that all users and institutions obtain prior authorization or licensing before procuring or using radioactive materials. Spill and accident management. In case of minor spills, e.g., a few micro C, absorbent paper and decontamination solution are used with gloves, while contamination levels are monitored using Geiger counters or liquid scintillation devices. For major spills, e.g., involving MCI amounts, the area must be immediately evacuated, the RSO informed, and the area cordoned off. Personnel potentially exposed should undergo bioassay or medical evaluation, and all findings are documented for regulatory review. Fire or flooding involving radioactive areas requires special handling, as these can spread contamination. Fire departments must be alerted to the radiological nature of the lab, and emergency services coordinate decontamination procedures. Decontamination procedures. Personal decontamination includes removal of contaminated clothing, washing skin with mild detergent, and medical evaluation if internal exposure is suspected. Surface decontamination uses specific chemical agents based on the isotope used, such as citranox, EDTA-based cleaners, or even dilute HCl or ethanol depending on compatibility. Decontaminated materials are either labeled for decay storage or disposed of via authorized waste disposal routes. International guidelines and monitoring. International bodies like the International Atomic Energy Agency (IAEA) and ICRP issue standards for radiation protection, dose limits, and training. Dose limits for radiation workers are generally set at 20 millisieverts per year average OVE. Microscopic techniques are methods used to observe objects and structures that are too small to be seen with the naked eye. These techniques are essential in fields like biology, materials science, and medicine. Here's a quick breakdown of the main types. 1. Light microscopy. Uses visible light and lenses to magnify samples. Types. Bright field microscopy. Phase contrast microscopy. Fluorescence microscopy. Confocal microscopy. 2. Electron microscopy. Uses beams of electrons for higher resolution. Types. Transmission electron microscopy (TEM) gives detailed internal structure. Scanning electron microscopy (SEM) shows surface structure in 3D. 3. Scanning probe microscopy uses a physical probe to scan the surface of a specimen. Types. Atomic force microscopy (AFM). Scanning tunneling microscopy (STM). 4. Other advanced techniques. Cryo-electron microscopy, used for imaging biological molecules in near-native states. Super-resolution microscopy, breaks the diffraction limit of light, e.g., storm, POM. Do you want a deeper dive into one of these or how they're used in a specific field? You said. For CSIR net-oriented theory lectures and previous year questions answers with high-quality lecture ChatGPT said. Memory updated. Here's a detailed CSIR net life sciences-oriented theory lecture on microscopic techniques, including previous year questions with answers with years mentioned. Microscopic techniques, CSIR net life sciences theory lecture. Microscopy is a central tool in life sciences, enabling the visualization of cells, tissues, and macromolecules. The choice of microscopy depends on the nature of the sample, required resolution, contrast, and whether live imaging is needed. 1. Light microscopy. Principle, uses visible light passed through or reflected from a specimen and magnified through optical lenses. Types. Bright field microscopy, the simplest form where light passes directly through the sample. It's suitable for stained specimens. Phase contrast microscopy, enhances contrast in transparent specimens by exploiting differences in refractive index. Ideal for live cells. Differential interference contrast DIC microscopy, uses polarized light for 3D-like images of unstained cells. Fluorescence microscopy, uses fluorophores that emit light upon excitation. Widely used in cell biology to detect specific proteins or organelles. Confocal laser scanning microscopy, uses laser beams and pinholes to eliminate out-of-focus light, enabling optical sectioning and 3D reconstruction. Applications, localization of proteins, visualization of organelles, and live cell imaging. 2. Electron microscopy (EM). Principle, uses a beam of electrons instead of light, hence resolution is much higher due to the shorter wavelength of electrons. Types. Transmission electron microscopy (TEM). Electrons pass through ultra-thin sections of a specimen. Provides detailed images of internal structures at the nanometer scale. Scanning electron microscopy (SEM). Electrons scan the surface of a specimen. Produces detailed 3D images of surfaces. Limitations, requires vacuum conditions and fixed dead samples, preparation is time-consuming. Applications, high-resolution imaging of viruses, cellular organelles, and tissues. 3. Scanning probe microscopy (SPM). Principle, uses a physical probe to scan the specimen surface. Types. Atomic force microscopy (AFM). A cantilever with a sharp tip physically interacts with the sample. Measures deflections to construct topographical maps. Scanning tunneling microscopy (STM). Measures tunneling current between probe and sample. Only works with conductive surfaces. Applications, imaging biological macromolecules like DNA, membrane proteins, and cell surfaces at atomic resolution. 4. Super-resolution microscopy. Need, conventional light microscopy is limited by the diffraction limit, approximately 200 nanometers. Super-resolution techniques surpass this limit. Techniques. Storm, stochastic optical reconstruction microscopy. POM, photo-activated localization microscopy. Principle, involves sequential activation and precise localization of fluorophores. Applications, imaging molecular architecture of cells with nanoscale precision. 5. Cryo-electron microscopy, cryo-EM. Principle, samples are rapidly frozen to preserve native structure and imaged at cryogenic temperatures in TEM. Revolutionized structural biology, particularly in understanding the structures of large complexes like ribosomes, viruses, and membrane proteins. CSIR net-oriented key points. Know the resolution limits of each technique. Light, approximately 200 nanometers, TEM, approximately 0.2 nanometers, SEM, approximately 10 nanometers, AFM, approximately 1 nanometer. Understand the advantages, disadvantages, e.g., live imaging possible with phase contrast and fluorescence, not with EM. Identify components of each microscope and their functions. Applications in molecular biology, cell biology, and biophysics are frequently asked. Previous year CSIR net questions 1. CSIR net Deck 2018. Which of the following microscopy techniques would you use to visualize live, unstained cells in culture? A. Bright field microscopy. B. Phase contrast microscopy. C. Scanning electron microscopy. D. Transmission electron microscopy. Answer. B. Phase contrast microscopy. 2. CSIR net June 2016. A researcher wants to examine the surface structure of pollen grains. Which of the following techniques is most appropriate? A transmission electron microscopy. B. Bright field microscopy. C. Scanning electron microscopy. D. Fluorescence microscopy. Answer: C. Scanning electron microscopy. 3. CSIR Net Deck 2017. Which microscopy technique would allow optical sectioning and reduce out-of-focus light in thick specimens? A. Phase contrast. B. Bright field. C. Confocal microscopy. D. TEM. Answer: C. Confocal microscopy. 4. CSIR Net June 2019. What is the approximate resolution limit of light microscopy? A. 200 micrometers. B. 200 nanometers. C. 20 nanometers. D. 0.2 nanometers. Answer: B. 200 nanometers. Would you like the next lecture on self-fractionation and separation techniques or go deeper into confocal and super-resolution microscopy? You said. Visualization of cells and subcellular components by light microscopy. ChatGPT said. Here's a CSIR Net Life Sciences oriented detailed theory lecture on the topic. Visualization of cells and subcellular components by light microscopy. Microscopic observation of cells and their internal structures is fundamental to understanding cellular function. Light microscopy remains the most commonly used tool for this purpose, particularly for visualizing live cells and dynamic processes. Principle of light microscopy. Light microscopy uses visible light, wavelength approximately 400 to 700 nm, to illuminate specimens in optical lenses to magnify the image. The resolving power is typically approximately 200 nanometers due to the diffraction limit of light. The image formation depends on light absorption, reflection, and refraction by the specimen. 2. Key components of a light microscope: light source, usually a halogen or LED lamp. Condenser, focuses light onto the specimen. Objective lens, gathers light from a specimen and magnifies it. Eyepiece, ocular lens, further magnifies the image. Stage, platform to place the slide. Coarse and fine focus knobs, adjust the distance between the specimen and the objective. 3. Types of light microscopy for visualizing cells and organelles. 1. Bright field microscopy. Description: light passes directly through the specimen. Use: stain cells, commonly used in histology and microbiology. Limitation: poor contrast for unstained cells, limited resolution. 2. Phase contrast microscopy. Description: converts phase shifts in light passing through a transparent specimen into differences in light intensity. Use: visualizing live, unstained cells, ideal for observing organelles like nuclei, mitochondria, and vacuoles. Advantage: enhances contrast without staining. 3. Differential interference contrast stick microscopy. Description: uses polarized light to produce high contrast images with a 3D appearance. Use: detailed imaging of live cells and organelles. Advantage: improved resolution and depth perception. 4. Fluorescence microscopy. Principle: fluorophores absorb light at one wavelength and emit at a longer wavelength. Use: detection of specific cellular structures using fluorescent dyes or proteins, e.g., DAPI for DNA, GFP tagged proteins. Applications: localizing proteins within cells. Tracking dynamic processes, e.g., mitosis. Multicolor imaging of multiple organelles simultaneously. 5. Confocal laser scanning microscopy. Description: uses lasers and pinholes to capture images from a single focal plane, eliminates out-of-focus light. Use: 3D reconstruction of cells and tissues. Advantage: high resolution and contrast, used for thick specimens or tissues. IV: staining techniques to visualize subcellular components: hematoxylin and eosin, H&E, general cell staining. DAPI or host, nuclear DNA. Mitotracker, mitochondria. Phalloidin, actin filaments. Acridine orange, nucleic acids. Live dead stains, viability assays. Stains in fluorescent dyes allow specific targeting of organelles and biomolecules, enhancing both contrast and specificity under the microscope. V. Applications in cell biology. Visualization of nucleus, cytoskeleton, mitochondria, chloroplasts, lysosomes, endoplasmic reticulum. Studying cell division, apoptosis, membrane dynamics. Live cell imaging to monitor real-time processes. Pathological diagnostics and cytogenetics. Limitations of light microscopy. Limited resolution, approximately 200 nanometers, cannot resolve structures like ribosomes or macromolecular complexes. Photobleaching and phototoxicity in live cell fluorescence imaging. CSIR Net-oriented key points. No types of microscopy and their advantages, limitations. Understand staining methods and specific dyes for organelles. Differentiate between contrast-enhancing methods, phase versus DIC. Recognize when to use confocal versus standard fluorescence microscopy. Previous year CSIR Net questions. 1. CSIR Net Deck 2018. Which of the following microscopes is best suited to observe live, unstained mammalian cells? A. Bright field microscope. B. Phase contrast microscope. C. TEM. D. SEM. Answer: B. Phase contrast microscope. 2. CSIR Net June 2021. A scientist wants to study the subcellular localization of a specific protein in living cells using GFP. Which microscopy technique should be used? A. Bright field. B. Phase contrast. C. Fluorescence. D. SEM. Answer: C. Fluorescence microscopy. 3. CSIR Net Deck 2016. Which staining technique is commonly used to specifically label mitochondria in live cells? A. DAPI. B. Acridine orange. C. Mitotracker. D. Beamsa. Answer: C. Mitotracker. Would you like the next lecture on transmission?